Hello, I'm Robert Chemelewski, and I'm from the Dr. Mullet Lab. Unfortunately, I cannot comment on whether or not he is a descendant of Colonel Johnson, but that headshot has circulated the lab a few times. So I'm going to be talking about characterizing the epicuticular wax pathway in sorghum. To start off, what is epicuticular wax? Well, the epicuticular wax is the outermost layer of the cuticle. Because of this location, it is the first layer of defense the plant has against anything coming from the atmosphere, whether it be a biotic or abiotic threat. The epicuticular wax layer is described as being primarily composed of waxy components and some terpenoids, but for this talk, we're going to be focusing solely on the polycosanols and aldehydes. And what's more important about the wax pathway is we really don't know how the crystals form, and they form rather beautiful crystals. All we know is they're there, they're stable, and they seem to be important. As a quick example, here is actually a photo of the wax crystals formed on internode 6 of the sorghum plant. And while there are many different morphologies, I'm just going to be calling your attention to 3 of them. The tubules, which are these long filaments here, the wax platelets or scales, which are these kind of fin-like shapes, and the wax plate, which is a solid structure that underlies all of this. Now, sorghum has been studied for many years because it accumulates such a thick wax layer, and it's thought that it plays an important role in its drought resistance and its tolerance of many forms of pests. But most of it has been done on grain sorghum, which, as you can see, is a tiny little plant. So they focused on the leaf and leaf sheath, as that's where all the biomass was that wasn't being harvested. I work with bioenergy sorghum here on the right, where we have engineered the plant to accumulate as much hydrocarbons inside the stem as possible. And by doing so, we've increased the length of the stem. And as such, we've increased the surface area of the stem, making this wax actually a relevant compound. And a lot of people ask me, well, how much wax is there? So on the left is a non-deconstructed bioenergy sorghum. And as you can see, there are thick wax flakes all up and down that sheath. Well, I deconstructed the plant, and here's an internode on the right. And I intentionally cracked the wax plate so you can get an idea of how thick that is. And it's 3 to 4 times the wax load of the sheath. So it's significantly higher levels. Now, our lab has done a lot of work over the years trying to characterize how the internode matures and develops. And during its vegetative stage, about every 4 days, a new internode is laid down, that becoming internode 1, what was internode 1 becoming internode 2, so on and so forth. Internode 1 and 2 are both largely rapidly dividing tissue. And when we get to internode 3, we have the first layer of fully matured cells. And by internode 4, all growth has stopped, and it's now only matured cells. So my hypothesis was right here at this mature zone is where we're going to see the first wax morphologies appear. My thought process being the cutin is no longer being remodeled, so epicuticular wax can be secreted. So just to go ahead and cover my bases, I started at internode 1 and saw the rapidly dividing cells. We have a rough surface. Going to the top of internode 3, we start to see tubule formation. So this little S here, and we can see it starting to form. That hasn't quite emerged yet. And then if we go to internode 4, all 3 morphologies I pointed out earlier, the tubules, the scales, and the wax plate are visible. So with this being done, I wondered if we could identify the genes responsible for it. Fortunately, the pathway has been identified in Arabidopsis previously, so I was able to do phylogeny and homology to see what genes were responsible. And as such, I created a list. Here's a shortened form of it that I've broken down into the fatty acid elongation pathway, the alkane forming pathway, the alcohol forming pathway, and transport. So since we saw the wax tubules starting to appear on internode 3, that's where we looked first to see what is being activated there. And what we see mostly is fatty acid elongation and CER4, which is known to make primary alcohols, and a little bit of the wax transport pathway is starting to turn on. So that leads me to believe that those tubules are actually fatty alcohols. So then what's the rest of it? Well, we go to internode 4, we see fatty acids turned on even more. Aldehyde production is increased substantially, or transcripts of aldehyde, CER3, and WSD1 has activated, but the wax transport pathway is really highly expressed at this point in time. So it's pumping out the wax as fast as it can. But something stood out to me, and some of the eagle-eyed viewers among you may have noticed this, is there's high expression of CR3 and CR1 in internodes 1 and 2, and the elongating part of internode 3. That didn't line up with anything I saw, so I was kind of curious what what's going on there. So CR3 and CR1 being co-expressed should make a alkane. So I turned to GC mass spec to test if these patterns are being actually expressed. So what I noticed was nonocosane, a C29 alkane, was being produced in internode 2, but I wasn't seeing any wax crystals there. And then when we went to internode 3, where we saw CER4 being expressed, which we expect to produce a long-chain fatty alcohol, we see the waxy alcohols appear, and that coincided with the tubule formation. And then when we went to internode 4, where we saw CER3 being highly expressed, and we were predicting aldehydes, we see aldehydes start to appear with all the other wax morphologies appearing. And then if we continue to go down the plant, like let's say internode 25, one of the first internodes that are produced, we see that the waxy alcohols and aldehydes have accumulated to substantial levels. If we look at an SEM, we find not only is the wax plate thicker, but those tubules are a lot longer. So with that being determined, all we have is what we think is producing the wax. The question is, how is this pathway regulated so we can try to engineer it? We weren't able to get high enough enrichment until we reached out to the people at IMSL to do some laser dissection microscopy for us, where not only can we show, yes, these genes are expressed almost entirely in the epidermis solely, but this gave us the enrichment to make a regulatory network. This is a relatively simple network compared to some of the ones people have presented today, but it's only 626 genes with about 1,400 connections in it. But the first thing we noticed was all of our wax biosynthetic genes fit into those 2 clusters. To further simplify that, we went ahead and pointed out the connections that were being made. Most of these transcription factors have been previously identified as part of the epicuticular wax biosynthetic pathway in Arabidopsis. But what we did notice for the starters that was interesting was MIB36 connected to the suberin biosynthetic pathway, which we were talking about earlier today, and MIB60, which has been connected to the epithelial, but never to epicuticular wax biosynthesis. Both of these are excellent genes to further investigate as they have a high number of connections to the pathway, or predicted connections to the pathway. So this got us to our model of where these rapidly dividing tissues are producing no wax crystals, but are making alkanes. My hypothesis is simply to smooth out and fill out holes in the cutin. Then the wax tubules start to form. The scales fill in under while the tubules elongate. More and more scales, oh, there it goes. More and more scales are produced until the wax plate forms. And that wax plate will continue to thicken as the plant grows, and those tubules will continue to elongate. This explains 90% of what we were observing, but what I haven't spent a lot of time talking about is where are these tubules coming from? It's been previously noted that these papillae, which are about a 2 micrometer in diameter formation, are the source of the wax tubules, and I see that they're the source of the wax tubules. But one thing I noticed is scale-like wax accumulates around these tubules prior to the tubule eruption. And if I try to do hexane washing, they hold these scale-like wax molecules right next to that surface, and it takes a lot of effort to get them off. So I started to think that there was something interesting going on there, and that's what I've been working on for the last year. And what my thought process is the underlying cell, which is called the cork cell, is actually producing a specialized cell, which is actually producing these wax tubules, primarily consisting of waxy alcohols, and that these papillae are a secreted formation that occurs into the cutin because I can see the cork cells prior and after the papillae formation, and there's some structural modifications that I can see to that cell as well. All right. As I said, IMSL was kind enough to do our laser capture microscopy, and the JGI has sequenced all of these samples for us. And with that, I'll be willing to take any questions.
Thank you, Robert. Is there a question or two for Robert? Anybody? Well, Robert, what's going to happen with the wax cells? So the plants are using these for defense effectively, and that from that perspective. So what are you going to do if you figure out what this pathway looks like? What's the application as you look at bioenergy sorghum?
All right. So what I normally like to talk about is how valuable these molecules are. So these are C28, C30 carbon chains, very long, and they have a large number of industrial applications. There are some plants that have been exclusively harvested for their wax, like Carnauba and Candilla, but the only grass that has been harvested for wax so far has been sugarcane. And as we all know, that's not the primary product. I believe that this could be a value-added product to sorghum to make bioenergy sorghum more sustainable as we continue to engineer them to accumulate more that particular wax.
Okay, well, thank you. Thanks, Robert, again. Appreciate it.